Cornell, his master's in epidemiology and his PhD in molecular and cellular biology. Those are done right here at UW. He worked at Fred Hutch while he was in Seattle during that period of his training. And then his postdoc was done at Columbia. And he's now an assistant investigator at the Pacific Northwest Research Institute. Please join me in welcoming Michael Metzger.
Thank you very much, and thanks for inviting me. It's fun to be here and talk to you all about contagious cancer in clams, and I'll mention some cockles and things as well. I want to talk to you and start off by introducing the idea of contagious cancer. So as we just heard, cancer is not normally contagious, not in the way that I'm talking about here for these contagious cancers. So there are infectious agents that we know of that can cause cancer in humans. See if I can get an audience participation. Does anyone have any idea, any examples of an infectious agent that can cause cancer in humans? Yes, that's the sort of biggest one that we know of and know really well. HPV can cause cervical cancer and other cancers in humans. But in that case, it's the cancer, it's the virus that jumps from one person to another. And then when it causes cancer, it triggers oncogenic transformation of each person's cancer cells. So the virus moves, but the cancer cells stay within each person. What we're talking about is not a virus moving. We're talking about the whole cancer cell itself moving from one animal to the next or one person. So it doesn't happen with people, but it happens in animals. And like Chelsea said, there are three main types of this. There's the cancer in dogs, one in Tasmanian devils, and then we find them in clams and other bivalves. So I want to talk about the first two that we know about that were known initially. First is this Tasmanian devil facial tumor disease. It's a solid tumor on Tasmanian devils that's actually spread by biting. When the animals bite each other, they will transmit cells that will engraft into the wounds of the other animal, and that's how it spreads through the population. It's actually about 20 to 30 years old. This is a really relatively young cancer lineage. And actually, there's been two separate independent lineages that have arisen within Tasmanian devils within this time. And so that says that if the conditions are right, you can actually have this generation of a transmissible cell line occurring multiple times. And this really is threatening them with extinction. Something on the order of 80% of the entire population of Tasmanian devils have been killed by this disease in the last couple decades. The other contagious cancer that's been known is the canine transmissible venereal tumor. This is also a solid tumor. It spreads sexually, so it's still spread through direct contact. And whereas this one is only 20 to 30 years old, the molecular clock analysis of this shows that it's 4,000 to 6,000 years old. So this means the original dog that had this first tumor was 4,000 to 6,000 years ago. And these cells have been essentially a cancer parasite of dog populations around the world since then. And this is actually found all over the world in feral dog populations. And it's basically on every continent except Antarctica, I believe. All right. So now I'm going to introduce you to soft-shell clams. These are the first species we ever looked at. They're not as photogenic as dogs or devils. So we're looking right there. That is the hole in the mud that when you dig that up with a shovel, you see the clam here as sort of a cross-section of that hole where you can see the hole here, and there's a little siphon sticking up through the mud. They kind of look like a smaller version of a gooey duck, if you're used to that sort of thing. The shells don't close all the way, and they have that little siphon. Now, these soft-shell clams, they're predominantly found in New England. There's sort of the classic steamer clams. Here's what they look like on the lab bench, and here's what you look like when you get a picture from the Food Network. But I started working on these because of this cancer that was known to occur in these clams. This is fatal leukemia-like amplification of cells in the hemolymph that then spread through the other tissues. So here's a picture of normal hemocytes. When you put them on a dish, they adhere to the dish and spread these pseudopodia out. This is what the cancer cells look like. It's a totally different morphology. They're these rounded refractile cells, and they really massively amplify within the hemolymph. This is a normal, a few drops of hemolymph in a well. This is diluted 1 to 100 because when you initially put the hemolymph in, it was so cloudy, it was just stacks and stacks of cells. Then they eventually kill the animal. Now, this disease had been reported since the 1970s in New England and Canada, and had led to some pretty severe outbreaks that caused a lot of mortality, but people didn't quite know what the cause was. A viral cause was suspected, but nothing had been found. And I was doing my postdoc in a retrovirology lab. And a marine biologist who'd studied this brought some samples and said, can we find the virus that was causing it? So I had an initial project trying to determine, is clam cancer caused by a retrovirus, trying to find the virus that was causing this. So I'm going to sum up a year or two of work as my postdoc in the next slide. And yeah, there was no virus, which was confusing to us for a while. But then we started thinking about different alternatives for this. And one of the wilder ideas was that this could be a transmissible cancer. So we eventually actually tested that theory after some preliminary evidence was suggesting it. And so we had to come up with an assay for determining whether a cancer is contagious. So to do that, we used a number of different genetic markers. And so what I'm showing here is actually a microsatellite, which is actually commonly used in forensic analysis with humans. But population geneticists have developed a bunch of different primers for microsatellites in clams and many other species. So what I'm showing here is one particular microsatellite locus. So microsatellites are small repeats like GT, 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 and they vary in the number of repeats in different individuals. Some will have like 20, some will have 22, some will have 24. And you can use primers flanking that repeat region, and then you can get the size of that band, and you can determine polymorphism in different individuals based on the size of that band. So in a normal individual, like these three on the left here, if we look at the band that amplifies from the tissue and the hemocytes, you can see that they match within a normal animal. And we can see that there's differences between these three animals. So that's what happens in a normal animal. You see differences between the animals, but within each animal, they're the same. In contrast, when we look at these samples of animals that had the cancer, the solid tissues and the cells we got from the hemolymph had a totally different genotype. There's a totally different band here. And notably, when we looked at all the different animals, the cancers all had the same band. So when we look at a number of different loci, this was 10 loci, and we built a tree, we can see that the normal animals, the genotypes are these black filled in dots around here. The tissues, the solid tissues of the diseased animals are these open red circles here. And so they're all very diverse. But when you look at the cancers, all of the cancers look like they originate from a single common point. And so this was really telling us that the genotype of all the different cancers from all these different animals, from Maine and New York up to Prince Edward Island in Canada, all came from the same individual originally. And we can look at some mitochondrial sequencing, and we saw some SNPs that were present in all of the cancers, and then some that were present only in one sublineage or another. So really through all these genetic tests, we were able to determine that cancer from Canada all the way to New York basically arose from a single clam that had this original cancer. So this cancer in clams was contagious. We basically use that strategy on these clams and other species I'm going to talk about, where you really look at, and within a normal individual, the hemocyte genotypes match the individual genotypes. But when we look at these animals with the disease, their host genotype does not match the genotype of the cancer, and all the cancer genotypes will match each other. And that's the main signature of these things. And I just want to put this here for the scale of the kind of spread we're actually talking about. And it looks like these are all sort of the one sublineage, and then we see a different sublineage around here in Prince Edward Island that seems to be relatively geographically separated. So now instead of just two of these types of transmissible cancers, we have the third one, bivalve transmissible neoplasia in softshell clams. So we have a whole bunch of questions about how this spreads, how it works. We knew initially in these softshell clam populations, there were these really severe outbreaks where upwards of 90% of population would die. We now know that the disease is still present in those populations, but we don't see the same really severe outbreaks. So we wanted to know, is bivalve transmissible neoplasia fatal in populations now, particularly looking at these softshell clams? And so we can use qPCR to really quantify the cancer-specific DNA. So we can look for markers. Again, this is sort of the neoplastic cells, healthy hemocytes. This was done largely driven by Rachel Gersh, who was a tech in my lab and is now a grad student in University of Oregon, where we have qPCR primers. We have two different primer sets. One is specific to this transposon insertion site, which was a marker that is very specific for these cancers. And then another was this control region that is right nearby. So we have primers that will amplify every single allele that we've found in these clams so far as a control. And then we have this primers that will amplify only the cancer-specific DNA. It actually only amplifies one of the cancer alleles, so that, so for example, in a healthy animal, the control primer pair will amplify very well. The cancer primer pair will get nothing. There will be no amplification. In a pure sample of cancer, we have the control showing a very high amount of amplification. And then we have this cancer quantification at about 50% of the total, because they have two alleles from starting from a diploid organism, and there is the steamer insertion site in half of the alleles. So that's why we reach 50% here. But we can use this qPCR assay to quantify disease progression over time. And so what I'm showing here, where we took about 20 animals that were diagnosed initially with sort of low levels of cancer. These were animals that we acquired from the wild. So we have 21 here that had low cancer at day 0. And then we had a control set of 39 animals that were undetectable for cancer at day 0. The first thing you can see is that undetectable doesn't mean the cancer is totally absent. We had a couple where they started out with undetectable levels of cancer, but they actually had cancer that was unable to be seen originally, that grew out over the course of this experiment. This also showed us that this actually takes quite a while to progress. This is weeks, so this is about 20 weeks here. So this they have these low levels of disease that can remain undetectable for a while and then grow out. In contrast, when we look at this low cancer, we see a lot of progression, and all these dots basically end in death of the animal. So we learned about the long incubation time. We also learned that there's a lot of variability in cancer progression. We're not seeing a very smooth, everyone progressing the same plot. So if we take these 20 animals and we sort of split them up into the patterns we see really 3 different patterns in those 20 animals. We see the main thing we'd sort of initially expected, progression to death, which all of these, they start low, and they progress up, and then the animals die. And we see this sort of progression and then death. And that's about half of the animals. But what we didn't really expect to see, we have a long-term non-progression in about the other half of the animals, where they've maintained this cancer at a very low level over weeks and months and even years. And then the other thing that we've seen in a few rare cases, what we're actually terming regression, where the cancer can grow up. In this case, it was more than 25% of the cells in the clam's blood in the hemolymph were cancer, and then it regressed. It was able to clear those somehow. And that is a pretty rare case, but we do see examples of this regression. And we have a big question in the lab: what is the genetic mechanism of this resistance? And we really don't know. We're just really initially at this stage of identifying the phenomenon and showing that there really is some sort of resistance to these cancers, but we don't know at this point what it's going to be. All right, so we're adding on to this a little bit. So the transmissible cancers in devils are fatal, and in dogs, they can regress or be treated, but in the bivalve transmissible neoplasia, we found that they are often fatal, although there is some regression, and it appears that there is evolution of resistance in these populations. Another thing I want to talk about here a bit is how does this actually spread? We know based on the genotypes that it has to be a transmissible cancer, but it doesn't have the direct physical contact of Tasmanian devil or dogs. These are basically sedentary clams. They don't even touch each other. They're usually separate in the water. And so we had the hypothesis that these cancer cells must be released from one animal and being engrafting in the other animal. But we wanted to test to see whether that was really a feasible idea. So we have a couple different tests of this. Again, a lot of this was done by Rachel. And that really, in order to go from one clam to the next, one of the key parts of this is that the cancer cells have to survive in seawater. So she looked at their survival in different types of conditions and found that they actually survive very well in the sort of middle range of salinity, which is what you see in most seawater in most of these regions. If you put the clam cells in low salinity, they really quickly die. They pretty instantly die in sort of a more freshwater environment. But they do survive quite well. So this is showing survival at only for 4 hours in seawater. But if we take the salinity conditions under which they work well, which is sort of standard seawater conditions, we can actually look longer. And that's when we start seeing that there's actually an effect of temperature as well. So now this is, instead of 2 hours, we're looking at days. And we can see that at different temperatures, each one of these colors is a different temperature, we actually see survival for days and even weeks. When we do the lowest temperatures, 10 degrees, 4 degrees Celsius, we see sort of a half-life of these cells of 2 to 3 weeks in artificial seawater, at least. So I think that really justifies our idea that these things must be transmitting through the seawater from one animal to the next. In addition to surviving in the water, we wanted to show that they can be sort of released by one animal into the water. So we've used eDNA, so environmental DNA. This is a place where you can actually take samples of seawater, extract DNA from it, and we can use those same qPCR primers I talked about before to identify cancer-specific DNA in the water. And so we have those same primer pairs. If we take water from clams that are in tanks, here we just have a pilot experiment where we have one healthy animal here. When we look at the eDNA, we can see that when we use our control primers that will amplify all softshell clam DNA, we can see a little bit of softshell clam DNA in the water, but we see no cancer-specific signal in those cells at all, in the water from those cells at all. Whereas these are two different animals that are highly neoplastic, and we can see that there's an enormous amount of cancer-specific DNA that can be detected in the water in the tanks of these animals. So we think that these animals are definitely releasing cancer-specific DNA into the environment, and we think this is in the form of viable cells, and that's how these cells are transmitting from one to the other. We can even look at eDNA from wild sites. So this is a couple sites in Maine where we can actually collect seawater directly from the environment and use these qPCR methods. We're pretty close to the limit of detection, but we can see a positive signal for the cancer-specific DNA, even in the environmental samples. And we're hoping to sort of make that more sensitive and use that even as a proxy for understanding transmission of the disease through the environment in the future. And when we look at the cancer cells being released by the animals, I showed before just two pilot examples of the animal that were heavily diseased. We took all the animals from that longitudinal progression I showed you before. So all the animals where they were either progressing to disease or maintaining it at a low level, and even the regression. And we tested for eDNA as those things were occurring. So all these are different points from a whole bunch of different animals merged together, with on the x-axis here, it's hard to see, is the percent neoplasia. So 0 means there's no cancer detected, all the way up to sort of 100% over here, 100% cancer. And this is the eDNA that's detected, and this is actually on a log scale. So there's actually a pretty wide variety of this eDNA that can be released. We essentially see almost no release of cells at the very early stages of disease until we hit about 25% of the cancer in the hemolymph, and then we really see it spiking up. And so it's really sort of the release of the cancer-specific DNA really tracks with the progression of the disease in the animals. Yeah. All right, so we really think that we have a lot of good evidence supporting this transmission through seawater. And so we think that is what's happening and allowing these cells to go from one animal to the next. So the next question I wanted to move on to, now that we know that it does exist and that it is able to transmit. So there's a whole lot of questions about these that we don't know. How old are these cancer lineages? How is it evolving, and what changes are occurring in the cancer itself? And so step one for that is we have to build a chromosome scale reference assembly. These are soft shell clams, and so we really have very minimal genomic resources. So there's the super detailed version. I won't go into all this. High molecular weight DNA, PacBio, Hi-C. This was actually done probably 3 years ago now, so probably the technology that you would use is entirely different by now. But in the end of it, we ended up with this boring graph down here that says we have got to 17 scaffolds. And so that means we got to a chromosome scale. There are 17 chromosomes. We got to a chromosome scale reference assembly here with pretty high completeness based on Busco. So if anyone wants to talk genome assembly, happy to dive into the details there. There was one previously published that was a little bit, has a much higher number of scaffolds. We actually had a first attempt where we used 10x Chromium reads and their platform, but that was just a complete disaster. And so the PacBio long reads with Hi-C was the way to go here. And then once we had that, we could really start analyzing the cancer genomes using short reads of the cancers and comparing them to the reference genome we had. And this was really driven by Sam Hart. He was a graduate student in my lab from the MCB program, and he just graduated, and this paper should be out next week. So initially, one of the first things we can do is look for single nucleotide variants in these cancers, and we found quite a few. So there's a huge number of mutations that could be detected in these cancer cells. So here we have samples from Prince Edward Island and samples from USA, Maine, and New York, and on a tree with the healthy reference clam genome shown over here. So this really long branch here, there's 2.3 million variants here. And now that's a bit misleading here because there's actually a mixture of the founder variants and the somatic mutations. So these aren't, in this branch here, that's not just the changes that happen in the cancer, because this is compared to a healthy reference, which is a different individual. We don't have the genome of the individual that started this cancer. So all the differences that are between the healthy reference clam and the original founder clam are mixed in here, as well as the early somatic mutations. But one of the really interesting things, because we have these two subgroups of the same cancer, we know that these SNPs that are here, SNVs that are here and here, those are somatic mutations, because those have happened since this lineage became a cancer. So we can actually use those, and we can look for characteristics of these mutations that occurred during the cancer. And we can use look for what's called a mutational signature. So this is looking at all the different types of mutations that will occur. So C to A, C to G, and so on here. And then each one of these bars is actually looking at the trinucleotide contacts. So that's not just C to A, but that's sort of ACA going to AAA. So it's each bar is one trinucleotide context having the mutation. And if we look at the top bar here, that's all the mutations in healthy animals. And then these are all the somatic mutations that occurred within the Prince Edward Island sublineage and the USA sublineage. And what we see is that there's this sharp peak over here, and there's a really specific signature of mutation that we see in these cancers. It looks somewhat similar to some mutations that occur in human cancers that make us think that it's likely due to a error-prone polymerase that's gone out of control in these cells for some reason. You can actually extract that out, and so this is the sort of cancer-specific signature of mutations here. And then Sam went further, and because we had this signature, we can start looking at the samples over time and the amount of mutations for each signature. And this mutational signature appeared to be relatively clock-like. So if we take the samples, we can plot a line on the number of mutations of this type that are occurring. And then we can also backtrack that to get the age of the most recent common ancestor. And there are some pretty big confidence intervals on here, but based on the signature S, as well as another signature that we are able to extract here, signature 5, it looks like the most recent common ancestor of those two sublineages was about 300 to 400 years ago. with sort of an early limit of it had to be sort of at least 200 years old. And then looking at these signature S mutations in that trunk, we can push that back to the original origin when it likely came from a clam, was even earlier than that. So we can really take home the message here that this clam cancer, this lineage, was over 200 years old. So the original clam that had this cancer as a sort of classical cancer would have been more than 200 years ago, with potentially far older than that. So we also looked at a number of other types of mutations in addition to just single nucleotide polymorphisms. We knew from sort of these crude analysis of DNA content that the genomes of these cells were roughly tetraploid. So the assumption in the field is that there had been a genome duplication where it had gone from a diploid cell to a tetraploid cell. But when Sam started looking at the copy number across the whole genome, so we have all 17 chromosomes listed across here. We have on the top in black, we have a healthy clam. So this is ploidy level looking at the read depth across the whole genome. Healthy animals, it all basically lines up at 2. So that means healthy animals are diploid, which is what we'd expect. But when we look in these Prince Edward Island and USA samples, this is really scattered all over the place. This isn't just a genome duplication. This is really an enormous amount of variation, high copy number, low copy number, and breakpoints throughout every chromosome here. And this isn't just a single sort of chromothripsis type event, which is an event where your chromosomes all shatter and rearrange at once, because this is continuing to occur. There's a large number of these rearrangements that are in common between these two samples, but if you look specifically, there's also PEI-specific rearrangements as well as USA-specific rearrangements. And we can actually see the profile of these copy numbers are different in these two lineages. So we're seeing huge amounts of structural variation and continued variation over time, which we really weren't expecting to see initially. There's other types of genome instability here. I'll just briefly touch on 2 of them. One, I mentioned a little bit that where there's a retrotransposon called steamer, which is a selfish element that repeats itself through the genome. We can identify the insertion sites, and we can see that sort of this is a transposable element that only has sort of less than 2 copies in the normal animals in black here. But in the samples from USA and Canada, we see over 100, almost 200 that are in common in all of them. And we even see one of these subbranches that has a burst of amplification of these retrotransposons. So these are more genomic changes that are occurring. This was just one particular transposon. Sam looked more high throughput at all the different transposable elements that could be detected in the genome, and looking at comparing their presence in the cancer cells to normal cells. And we really see this skew out here where all these dots are a different type of element that is more highly amplified in the cancer than it is in the normal cells. So we're really seeing a large number of different elements that are amplified in these cancer cells. And the other thing I wanted to talk about was mitochondrial genomes. So right here we have the mitochondrial genome map. It's about 17 kb, and what is being plotted here is the read depth across it. One of the things you can see is that there is something strange going on here. And instead of this sort of depth that is normalized to 1, it spikes up with a huge number of reads. What we found is that there's a region that is expanded and repeats are occurring. We quantify this. We see that in healthy animals, it's actually repeated. We see 3 copies or 4 copies of this repeat region in healthy animals. But when we look at these cancers, we see many more copies of this. So that this mitochondrial genome keeps amplifying during the evolution of these cancers, and it's continuing to change. So the conclusions here. So disseminated neoplasia is a transmissible cancer, and these bivalve transmissible neoplasia cells, they spread through the water. And we've really shown with this genomic analysis that this thing has survived as a pathogen for more than 200 years, really despite this pretty unexpected widespread instability and continuing mutation through a number of different types. I want to briefly talk, so that was all focused on soft-shell clams, which was really the first species we've identified this type of cancer in. But I want to go beyond that and look in some other bivalves. So Similar diseases that were called disseminated neoplasias have been found in many bivalve species all over the world, in many different geographic regions. In softshell clams, this was identified as early as the 70s, and so there's a number of reports from many different species in the order of 1 to 2 dozen species that have occurred over the time. So we started looking at samples that were identified as this disseminated neoplasia in a number of different species. So the first study we did looking at more than initial clams were through collaborators in Canada, looking in mussels, and collaborators in Spain looking at cockles and these carpet shell clams. And really, in each case, what we found is that the disseminated neoplasia was a transmissible cancer, and that it was a new transmissible cancer in each case. So in mussels, we found a mussel-specific transmissible cancer spreading in those populations. In cockles, we actually found 2 separate lineages spreading in the same population in Galicia. And then in golden carpet shell clams, we found something that was a little bit odd. We sequenced the cancer, and we sequenced the hosts, and we first looked at CO1, which is the easiest thing to look at. This is a conserved region in mitochondria that's often used as the sort of barcoding primer in a lot of genomic studies. And we saw that there weren't just 1 or 2 SNPs that were different. It was more like 10% different. It was a lot more different than we were expecting. And so we put it on blast and found out that the cancer, not only was it a different individual, it was actually a different species. So this was the first time we saw a cross-species transmission of cancer, where the cancer in this species came from this pullet shell clams. They are closely related. They're in the same family. They were in the same genus when I started, but taxonomists do their thing and I stay out of that. So they are closely related. And since then, we have seen other cases of transmissible cancer that jump species, and it's always relatively closely related species where we see this. But one of the interesting things about this is that this cancer spreads within this population, but these 2 clams are in the same beds, in the same areas. And this one has never been reported to have any disease. So it looks like the original host species is now resistant to this cancer that arose from it, even though it's being exposed to it in the seawater. So that again shows that there's sort of this evidence for resistance in these clams that we don't understand yet. Another one I wanted to mention, so I talked initially about our first study where we found a cancer in softshell clams from British Columbia, which we're sort of giving it away by labeling it BTN number 1, that we found more cancer lineages in the species. When I first started my lab, I had 2 different projects. I had 2 techs. And we'd collaborated with Nicolas Biern's group and Gloria Arriagada and Nuria Vasquez's group in South America. And so one of my techs worked on the Edulis samples and the other one worked on the Chilensis samples. And I was thinking they would each have a nice little paper describing potentially a new cancer. And then they got their sequences back and they were the same lineage of cancer. So the same lineage, which we're now calling Middle East BTN2, is in Europe as well as in South America. So the single lineage is actually spreading between different species of the Middle East genus and spreading essentially all over the world. An independent group has recently found that same lineage in the Sea of Japan and in Northern Europe as well. So that one really is worldwide. And so now, instead of really calling this a clam transmissible neoplasia, we really have to call it bivalve transmissible neoplasia, because this really is sort of a broad phenomenon that we're seeing many times over. So then we're seeing a lot of different neoplasias in different species here. So looking specifically here in Puget Sound, we want to tell you a little bit about one new project looking at cockles. So this is a different type of cockle species. The common cockle was the one found in Europe. But here we're talking about basket cockles. Now these are probably about that big in Puget Sound and on the West Coast. And these are an important part of the culture and diet of the Suquamish and other Coast Salish tribes. They've been dwindling in numbers. And so the tribal shellfish biologists, primarily from the Suquamish tribe as well as others, have been working with PSRF, this Puget Sound Restoration Fund, with the idea that we're going to make a hatchery to increase the numbers. But their initial health screening showed that they had a large amount of this transmissible cancer in at least one of their populations. And so then they reached out to us and we started this study of looking at these samples. This really matches the same kind of phenomenon we were seeing before of these sort of rounded cells proliferating within the hemolymph. And so we wanted to know, is this a bivalve transmissible neoplasia? So the short answer is yes, it was. We looked at a different marker because there were essentially no resources, genomic resources for this species available at the time. We looked at alpha tubulin, which is a conserved region where we were able to get primers that would amplify in these cells. And we're looking at 3 healthy examples here from a pilot set. And we just randomly number or make letters for the different alleles we find. So this one up here has the allele we're calling A and C. It's the same allele present in the hemocyte and the tissue. So this is the same thing we've seen before, where we are identifying different alleles in different individuals that are the same in all the tissues in normal animals. But we're seeing a difference in the genotypes that we're finding in the cancers and the hosts from those cancers. These, we actually see some variation, and we actually see this as a triploid region within these cancers, but we do see these common alleles with a small amount of mutation between them. So we know that this is another transmissible cancer. And we can build qPCR primers that are specific for this allele. And this is what we're seeing over here. In healthy animals, we see no amplification. And in these neoplastic animals, what we see is there's a huge amount of the amplification in the hemocytes, but a lot less of that cancer-specific allele in the tissues, which is sort of characteristic of these cancer cells that proliferate first in the lymph and with small levels of dissemination into the tissue. So that really confirms that what we're seeing is a sort of a cancer-specific signal in these cells, in these animals. And we've gone and surveyed a number of different sites, 11 different sites in Puget Sound, using qPCR as well as this hemocytology assay that was developed by our collaborator, Lauren Vandepa, in sort of University of Miami, as well as out here working with PXRF and Ben Arroyo. So we looked at this K allele, which was this cancer-specific allele, and we screened across a number of different samples, 30 samples from Samiamu. The first one that we found that was really the pattern we were expecting to see for cancer is this one, SM26. We have a very high level of this cancer-specific allele in the hemocytes, but a very low level on the tissue. So this is a discordance between the hemocytes and the tissues, and it was this allele that we thought was cancer-specific. The other thing we're seeing here, we weren't really expecting it, but what we're seeing is that there's this allele that we thought was cancer-specific, but is present at the same amount in the hemocyte and the tissues of these animals. And so what that is telling us is that what we thought was a cancer-specific marker, it also is a normal polymorphism in the population. And these are the genomic copies. So we're seeing that these are normal animals that just happen to have that allele in their inherited germline DNA. So those don't have cancer. This was really the only one that has cancer through this qPCR screen. And then that matches with what we've seen with this slide smear histology technique. These were the cancer cells, and these were the normal animals. We had one other odd thing in this set, that we had this other animal, SM6, where the cells really looked like they were neoplastic, but they were negative by this qPCR assay. So then we thought that maybe it was a second lineage, sequenced the allele from that sample, and then definitely we found that it was a cancer-specific allele, where there was a discordance between the cancer and the host genotype. And we can build a qPCR primer for that, and we can find that, yes, we can quantify this cancer-specific DNA in this second lineage. So this appears to be an independent lineage of cancer from this first one. So we can now see that even within this single population, there are 2 lineages of this cancer spreading. This was the Samiamu population. So right now, I've only been showing you the alpha tubulin results, and those are all pretty consistent, that we see these 2 main lineages. We see a number of different animals that have this BTN1. So far, we've only actually seen one animal that has the second lineage. but all of the animals that have this BTN1 lineage, they have these two specific alleles in common. Now, in contrast, when we look at the mitochondrial genome, again, this is looking at this mitochondrial CO1 locus, where we have some diversity, we see an unexpected pattern in that the sequences we find in the cancers are different from their hosts, but the sequences we find in the different cancers in lineage 1 are really all over the place. They don't match a single clonal phylogeny. And so what we think is happening here is that there's actually been horizontal transfer of the mitochondria. So this is evidence that the nuclear DNA of these cancers is a single lineage, but they have acquired mitochondria from their hosts or from other cancers or from some other source multiple times. And so the mitochondrial lineage actually doesn't look like it comes from a single phylogeny, even though the nuclear lineage does. And so this horizontal transfer is actually a phenomenon that has occurred multiple times within the canine transmissible venereal tumor. But we haven't seen a bivalve sample, bivalve species with this much horizontal transfer before. So it's really sort of an unexpected finding here. And I just want to put up the map here to show where these cancers have been found. Both of those lineage 1 and 2 were found in Semiahmoo, and then we found them in Penn Cove and Kiana Lodge Agate Pass area. We also found what is possibly a third lineage in Squim Bay. So we find these in multiple sites in Puget Sound, but they're not really ubiquitous all over yet. So we have a lot of future plans here. So we've found disseminated neoplasia in a lot of different species, including these basket cockles in Puget Sound. 3 lineages have been found. 1 is widespread, but the other 2 don't appear to be. So we don't really know why one would spread more than the other. And we know that these BTN, the bivalve transmissible neoplasias, can really have severe effects on aquaculture and hatchery plans. So we have a lot of ideas in the future and a huge number of collaborators on how this BTN will spread within a population, as well as how it spreads between populations. And we've just started a big grant in collaboration with all these groups to try to answer those questions. Just briefly, we are looking at a small number of sites many times a year to look for seasonality and temperature changes. And then we're also looking between sites, so once a year sampling a larger variety of sites to see if we can find out how the BTN can go from one population to another. So that's looking at both cockles and softshell clams. So cockles in Puget Sound, softshell clams on the East Coast. This disease is enzootic in New England, but we actually recently, just last year, found samples of softshell clams on the West Coast that had this disease as well. So we've collected the softshell clams from a number of different sites. These 2 sites actually have the transmissible cancer, and these do not. So they really have, even just in Skagit Bay, we have an area that has the cancer and an area that does not have the cancer. So we're really sort of interested in following this up and finding out how these things spread, if they spread in these populations. And this is sort of the brand new preliminary data that we're hoping to develop soon. All right, I want to thank everybody for listening. I have a really out-of-date photo for my lab. I just realized yesterday that I need to get another picture with all the new people, because most of these people have gone off to graduate school or postdocs. So thanks, everybody.
So, as the couple of these that have been aged, you talked about the age, all seem like they are more recent and is in evolutionary time.
Mm-hmm.
Is the thought that cancers are evolving faster now, or is there a mechanism where we think maybe there were cancers 10,000, 5,000 years ago, and somehow they died out and aren't detected?
Yeah, that's a good question, because they're really old if you think about the lifespan of an individual, but they're really young if you think about the evolution of life. And so really, if you think about sort of cancer in more broadly, cancer has really been a problem since the origin of multicellularity. As soon as you have cells working together, you can have cells that are cheating and don't work together with the common plan. And really there's nothing saying that these transmissible cancers couldn't have happened back then, and that there could have been early transmissible cancers even at the beginning of multicellularity. We're not seeing ones that are that old. We're not seeing things that are really ancient. So that may suggest that there's some sort of either lifespan to these things, or that they are sort of recognized and cleared from their hosts and they go extinct over time. We don't know whether there's any sort of intrinsic failure to them. So we were talking about the mitochondrial replacement, and one of the theories for why we see this mitochondrial replacement is that there could be this accumulation of deleterious mutations, sort of a Muller's ratchet of mutations that occur in mitochondria and maybe in other parts of the genome, that they can't clear, and it basically will continue to degrade unless it has sort of replacing mitochondria from the host. So they may be sort of inherently unstable at the broader evolutionary timescale, but we really don't know. It's a fun question.
I think I missed something fundamental. So the cancer cell, or a cell that becomes cancerous, has something messed up with it. It's ejected, circulates in the environment, makes contact with an uninfected animal. And then what happens? Does that cell then outcompete the animal cell, or does it get its DNA into the host cell?
So that whole cell will engraft somewhere in the other animal and start to divide and become a cancer in that animal without necessarily sort of transferring any DNA. Those whole cells will continue to divide, more like a parasite itself than like a virus that gets into cells. And so it really is, it's sort of thinking of it like in metastasis, you have a cancer from one location in your body where those cells will get into the bloodstream, float around, and then graft into a different place in a person's body. This is sort of the same metastatic phenomenon, but instead of going to a different place in the same body, it's actually going to a completely different body. And it's sort of like an interpersonal metastasis.
So then it outcompetes.
That it outcompetes the host in that new situation.
But then what's it's a hemolymph? What is that?
So the hemolymph in bivalves, it's the sort of innate immune cells within invertebrates. It's a phagocytic. There are some granulocytic subpopulations. So it is sort of an immune cell that the normal hemocytes can sort of disseminate through the animals and spread through the different tissues. So both our group and one other group that working on the cockles in Galicia did some transcriptomic analysis, and it does look like these cancers are most closely related based on their transcriptomic profile with normal hemocytes, so that the cancer probably did arise from a hemocyte, but we don't really know if there's, and we see the same phenomenon in multiple species, so there's probably something particular about that cell type that lets it be more likely that will turn into these cancers, but we don't know what that is.
You mentioned the three kinds in Tasmanian dogs and bivalves. Why isn't this type of cancer more widespread? Is there anything about it that specifically applies to those groups?
So I think the major barrier for these transmissible cancers, I would say there's two major barriers. One is sort of a route of physical spread. So you need some sort of physical spread for it, but that's something that can be evaded. So we have biting, and there's sexual spread, and then marine seawater transmission. But then the other major barrier is the adaptive immune system, and the MHC. So these are the self-non-self recognition systems that vertebrates have. So as we were saying before, when we were talking, so the same reason that if you got my kidney, you would reject it. Your body would recognize it immediately as foreign because the MHCs would be different. That's the same reason that you could not take cancer cells from me. Your body would immediately reject them as foreign. So the immune system and the self-non-self recognition system of vertebrates is very good at removing these things and keeping transmissible cancers from happening. That said, it happened in devils and dogs. And I think the biggest question in those is sort of how were they able to evade that MHC self recognition? And I don't really know the answer to that at this point. Something there could be population bottlenecks in Tasmanian devils and maybe in the initial dog populations where that happened. But yeah, we don't know that. I think that's a really interesting question. And then the other question that comes out of that is, why aren't there more in marine invertebrates? And I think there are. That's my thought. We only really found these in clams because you saw a massive outbreak in a population that was important for aquaculture. And people studied it and figured out that there was this transmissible cancer. So this sort of thing might be occurring all the time in environments, and we just don't know about it.
You know if non-cancer cells can't graft on as well?
Yeah, so if non-cancerous cells can engraft. We haven't done good experiments on that. I've done some pilot experiments where I just sort of take some hemocytes from one animal and stick them in another animal and qualitatively follow them using primers. And I can see that if you take sort of healthy cells from one clam and inject them into another clam, they are still detectable for at least a week. Whereas if you take cells from a different species and spike it into a clam, they're cleared within a day. We don't know. I have no idea what the mechanism is of that kind of thing, and I've only done that in a small scale. But that's a really interesting question, is sort of what are the normal mechanisms that are able to clear it, and can those cells normally engraft into others? So we don't really know the answer to that question.
You think it might be possible that resistance spreading that way as well?
Yeah, so well, resistance in that it goes from one animal to the next. It's possible. I would think that the resistance would be more likely to occur sort of based on just death of susceptible animals. But I think one of the things I think is my best guess for what the resistance is however cells, hemocytes will actually bind to the tissues, whatever that cell surface interaction is. I think that is likely to be the place where we see these mutations. So there might be a situation where whatever cell surface receptors on the solid tissue the hemocytes bind to, if those can mutate, then maybe the cancers won't be able to bind and engraft into tissues. But we don't know the answer at this point yet.
Okay, let's continue with questions outside in the lobby during TGIT. So please join us for that post